Hello, everybody. My name is Gilles Bouchard. I'm the chairman of the board for the CMTA, and I'm here with Catherine Forsey to introduce the STAR section of this afternoon and of the conference. We also have our first panelist with us, and we'll go right into the panel after introduction. First, I want to thank everybody involved. We've assembled an amazing panel of scientists and partners, and I want to thank everybody for their time, and also thank the team and all the volunteers that helped make this conference amazing. I was listening since this morning, and it's just a fantastic conference, a great platform. So there are a lot of people behind the scenes doing amazing work here. So thanks, everybody. I'm very excited about this STAR session. First, because we have some amazing people, and we're going to cover some amazing ground. But probably even more because it's such a great reflection of how far along we've come in the STAR program. For those who've been with us for a while, you know that when we started STAR, well, there wasn't much to get excited about in CMT research. And the whole idea of STAR was, let's make CMT attractive to partners, attractive to researchers, attractive to new technologies. And I think you'll get a glimpse today well this strategy is working. CMT has become an attractive ecosystem, an easy ecosystem to work with, and you'll get some idea of that today, and it continues to grow. There used to be a time where we had to beg people to come work with us, and I can tell you that we get calls on a weekly basis, on a monthly basis now, of researchers, companies who've heard about CMT, who want to work on CMT, and they tell us, remember, I used to say there's 5 things we've been working on to make CMT attractive, and they bring all those things up. There's great expertise, there's fantastic tools, there's fantastic models. There's been a lot of progress on the clinical side with biomarkers, and the one that always makes me happy, they say, you guys have a great reputation as being a good organization to partners with. So again, I think you'll get a feel for that for yourself. Don't take my word for it, but this is what STAR is today. And again, a lot of people have been involved with that. But the most important is, at the end, this is your program. Remember that the reason why we're able to invest so much money in STAR is why we're so efficient with the money and why we get those perfect ratings is because most of our money, in fact, all our money comes from you. So we don't spend hardly money on marketing and sales and events and all this fancy stuff that most organizations do. 90% of our money on average the last few years has gone directly into our programs. It's all thanks to you, because that's what we focus on. So keep that in mind. Keep that in mind that the whole strategy is about partnerships and making CMT more attractive. And then now I'm going to introduce Catherine Forsey, who joined our team this year. Has been an amazing addition to our team, and she'll show you the details. Catherine.
Thank you, Gilles. It's great to be here with you all today. As Gilles said, I'm new in post here at the CMTA. So I was recruited to the newly created post of Chief Research Officer in May, just this year, to oversee the CMTA's impressive and massively expanding research portfolio. And I get to work with our STAR Advisory Board, which is a 30-strong team of world-leading scientists, many of whom you're going to get to hear from this afternoon. We also work with the translational experts and CMT clinicians, and you'll be hearing from them too. So my first 4 months, it's just been an absolute whirlwind. I've just been completely blown away by the massive diversity of research that the CMTA is funding and the awe-inspiring knowledge of those who've chosen to dedicate their professional careers to improving the lives of people with CMT. And when I say people with CMT, that also includes me. I'm mother to an 8-year-old with CMT, sister, daughter to family members who all have CMT just like me. I also happen to be a research biologist. I wanted to be a scientist since I was 11 years old. My PhD is in biology, and I was studying gene expression, enzymes, and proteins for many years before I found out that I have CMT. I got my diagnosis just 4 years ago, and it was a surprise. And like all of you, I came across the CMTA when I was looking for information and support. I put my hand up to volunteer for Camp Footprint, as you've heard about in one of the earlier sessions, and I got to know the CMTA leadership team. And now here I am 9 months later, and I just feel like I have the most amazing opportunity to make a difference to the lives of all people with CMT. So my own family has joined your wonderful CMTA community. They're watching today, and it makes me immensely proud to be able to tell them and to tell all of you that there is hope. We are making progress. We are making real and significant progress towards effective treatments for CMT. My appointment to the CMTA represents the board's commitment to our strategy to accelerate research, which we call STAR. We're expanding our research, we're expanding our network of alliance partners, and we're expanding our global reach. You might have noticed from my accent that I'm British, so I'm based here in England, and that also helps to position the CMTA to work even more closely with our friends at the European CMT research associations, research laboratories, pharmaceutical and biotechnology companies that are spread right across the world. So it can be common for me to be on video calls with people in 4 or 5 different time zones. It's not surprising that we've got representatives of 30 countries joining us at this summit today. And it just goes to show that the CMTA has a global influence and a huge collaborative approach in seeking out experts who can support our mission. I'm really excited for you all to hear about the CMTA's funded research and the progress that we've made since the last summit. The Charcot-Marie-Tooth Association is at the forefront of CMT research. We've invested over $18.5 million just in the last 10 years, and there is more to come. I'm really excited for the treatments that are going to be developed in my lifetime and in my son's lifetime. And we're driving all of this through STAR. And I'm just so pleased that you've joined us this afternoon to hear about the work that your donations are funding. I'm going to share with you now what you're going to see this afternoon, so some details of the STAR sessions. I'll just bring up the agenda slide, and then you can have a look at what you're going to be seeing. So here is your afternoon, and this is a stellar lineup of researchers. We're so fortunate that they've all been able to join us. So we've got researchers, clinicians, pharmaceutical and biotechnology representatives. Straight after me is going to be the gene therapy panel, and that's introduced by Cleopas Cleopa and moderated by John Starren, who's going to be asking questions of our panelists. At 2:55, we're going to head into the drug development panel, which is introduced and moderated by Mark Scheidler, who leads the CMTA's Translational Expert Board and our Alliance Partnership Programme. Mark's going to be joined by some of our STAR Alliance partners, and they'll be sharing an update on their work and what we can look out for in terms of drug development. The final panel is our clinical trials panel, and that's introduced by David Herman and moderated by Tom Dubensky, who's from the CMTA Board of Directors. The panelists are going to share the current status of clinical trials for CMT and the advances that have been made in clinical trial development and delivery. Gilles and I will be back with you at 4:15 to close the panels, and we'll share with you how you can get involved in the CMTA's research and how you can work with us to support the development of new treatments for CMT. For now, you don't need to move. In a few minutes' time, right here in this room, we're going to have our gene therapy panel. I think you'll have a couple of minutes to grab a cup of tea before you join back with John, Cleopas and the rest of the panel, and they'll take you into that world of gene therapy. I'm just as excited as you are to hear what they've got to say. So thank you for joining us for this afternoon, and I will see you in a few minutes when the gene therapy panel starts.
Okay, welcome everybody in this gene therapy session. We have a group of specialists here that will also answer questions that are relevant for this topic. My name is Cleopas Cleopa. I'm a neurologist at the Cyprus Institute of Neurology and Genetics, and I do research on CMT neuropathies and gene therapies. And so I will give you a very brief introduction to the topic of gene therapies for CMT neuropathies, and then we will have the panel discussion. So one important consideration when we discuss gene therapies for CMT neuropathies is to understand that CMT neuropathies are quite heterogeneous. So we have many different genes that cause neuropathies, more than 100 different ones. These genes can have very diverse cellular functions, and therefore we have many different disease mechanisms. One important aspect is that these genes can either be very important for myelinating cells, the Schwann cells, and when they have mutations, we have demyelinating CMTs. And other genes are important for neurons and the axons. So when they are affected, we have axonal type of CMT. A second very important point that we need to consider is whether this mutation and this particular CMT type is caused by a gain of function, a toxic effect of the gene, or by a loss of function, because this will mean a different approach to treatment. Very briefly, when we talk about gene therapy in general, we mean the introduction of genetic material into people instead of drugs or using surgery for treatment, and it can take different forms. One of the approaches is to replace a faulty gene. We use that when we have a loss of function, so we have to replace the healthy gene, or in other cases where we have a gain of function or an excessive amount of the gene product, we use inactivating or silencing approaches. And finally, we also use gene editing, which is like a cut and paste approach. Sometimes it's only cutting, and sometimes it's cutting and replacing with a healthy section or a whole gene. In CMT neuropathies, we have particular challenges in terms of delivering gene therapies. One very important aspect is the access to the peripheral nervous system, which is not easy because the nerves are protected by a blood-nerve barrier from the circulation. And so we have to use different vehicles to deliver the gene therapies and different injection methods. We divide the therapeutics into non-viral and viral. The non-viral use usually small molecules like liposomes, nanoparticles, or antisense oligonucleotides that introduce the genetic therapies into the tissue of interest. And the viral approaches use viral vectors that are non-infectious, but we use them to carry the genetic material. The currently most commonly used is the AAV, or adeno-associated viral vector. And we inject these therapeutics in different ways to target the nerves, including lumbar intrathecal injection or intravenous, or directly into the nerve, or even subcutaneously for the antisense oligonucleotides. So there is already a lot of progress in the effort to develop gene therapies for CMT neuropathies. And you see here a diagram of the CMT neuropathies by frequency. Of course, CMT1A is the most common, is more than half of the cases. CMT2A, 1X, and 1B are the next most common. Currently, there are gene therapy developments for many of the CMT neuropathies, including the most common ones. And we use many different technologies to treat CMT neuropathies, including most of the ones that I already mentioned, the viral vectors, silencing or gene replacement, gene editing, antisense oligonucleotides, and also non-viral approaches. Most of these are actually tested for CMT1A, which is the most common and the most attracts most of the focus in research. Finally, I would just like to mention that gene therapies do have a long path to clinical application. So when we want to develop a treatment that will reach patients, we start with the understanding of the disease mechanism. Based on this, we design the appropriate gene therapy approach, as I alluded before. Then we have to test this treatment either in cell culture systems or in animal models. We have to provide a proof of efficacy, and the optimal dose needs to be shown. And also we need to evaluate for toxic effects. And then we move from the small animals, rodents, to big animals. This is a big step. Usually non-human primate studies are needed to validate the biodistribution and the safety before we are ready to apply, for example, for an FDA approval. At the same time, we need natural history data to tell us about the course of the disease, what we need to use in clinical trials as outcome measures, and how the disease progresses in order to be able to plan clinical trials. So all of this hopefully will lead to clinical trials that would be successful and allow treatments to enter the clinic. So this was my brief introduction. Thank you very much. And now I will stop share and allow my colleagues to also introduce themselves and start discussing.
Thank you so much. My name is John Sferrin. I serve as chair of the Scientific Advisory Board for the CMT Association, and I'm also at the University of Wisconsin. It's great to get this started off with Dr. Cleopa, who's really been a pioneer in this field. And we're also fortunate to have several pioneers in addition on this panel for question and answers. So I'd like to ask, you already have gotten to meet Dr. Cleopa, but I'd like to ask each of the panelists to introduce themselves in turn. Maybe we could start with Katrin.
Yes, thank you, John. Thanks, everyone. I'm very excited to be here. Kat Meyer. I'm a principal investigator at the Center for Gene Therapy in Columbus, Ohio, Nationwide Children's Hospital, and assistant professor at The Ohio State University. We have translated multiple AAV, so viral gene therapies, to the clinic, including last year a gene therapy for CMT2S.
Excellent. Luke, please go ahead.
Thanks again for the invitation. Great to be here as well. My name is Luke Judge. I'm an associate professor of pediatrics at the University of California, San Francisco. And I also work at the Gladstone Institutes in San Francisco, which is an independent research foundation associated with UCSF. And our interest is in using gene editing with the CRISPR-Cas9 approaches specifically for axonal CMTs. So CMT2E, 2A, and 2F are some of the ones that we've been already focusing on.
Diana, would you introduce yourself?
Sure. My name is Dr. Diana Barucha-Goebel. It's a pleasure to be here today. I'm a pediatric neuromuscular neurologist, both at Children's National Hospital in Washington, D.C., as well as at the NIH as a research collaborator. My work has been in gene therapy trials for one disease, giant axonal neuropathy, as well as supporting other studies. And some of my work has focused around immune modulation for gene therapy trials, looking with the intrathecal route of delivery for this particular study, but also looking at outcome measure development and how that supports trial readiness. So thank you for having me today.
Mike?
Yeah, thanks, John, and thanks to the CMTA for inviting me today. So I'm Michael Huang, and my role is I am the Chief Medical Officer at DTX Pharma, which is a small biotechnology company. We're based in San Diego, and our focus is on RNA therapeutics with a fatty acid delivery system. I am a clinician by training, and main focus that I'm a part of is really bringing therapies into the clinical trial space, bringing them from phase 1 to 2 to 3, and hopefully onto the market for patients who need them. So again, thanks very much for the opportunity to be here.
Excellent. So I think we'll just start with some general questions and ask everyone to chime in on this. So the first question is, what kinds of questions or issues do scientists or companies consider when developing a gene therapy for CMT? And from your perspective, what kinds of approaches do you think really hold the most promise for CMT? And so maybe we'll go in the same order. So Katrin, do you mind chiming in on that question?
Sure, I have an advantage. I get to go first. Well, certainly we need to know the gene and especially which cell types are affected. That will help us potentially select the route of delivery and then also the type of gene therapy, viral versus non-viral, safety considerations. And ultimately, we'll also have to think about, age of the patients that we can treat if it's a degenerative disease. Maybe we have to treat early. So I'm going to stop there and see if other folks have other things to add.
Luke?
Yes, I would have all of the same considerations as Catherine on my list. The way, one of the things we thought about a lot is the tools that we have and what those tools were already very good at doing, and thinking back to, as Cleopas introduced, those different categories and strategies based on the genetic cause of the disease. And so we try to pick the disease that we think that our tools could be most effective at intervening. With gene editing, I think there's an additional consideration specifically about the side effects and the risks when we're making a permanent modification to the person's own genetic structure. And so that carries an extra level of scrutiny that we have to do to be able to decide when these things are ready to test in people. And the other thing we think about a lot is how reversible the process is, which gets back to Catherine's question about the age that we might treat people is do these things have the capacity to reverse symptoms that have already been happening, or can they only sort of halt progression? And those are questions we're interested to answer.
Thank you. Diana?
Yeah, so kind of taking those responses to kind of layer on, maybe even thinking about translation into the clinic based on really enabling preclinical and IND enabling work. I think you want to think about, again, as Kat mentioned as well, what's that, for each given condition, what is the target population? Is there, for example, a very varied phenotype, meaning from maybe more severe early onset to slightly mild or later onset of disease? And do they have different trajectories? How do they change over time? Are you targeting both? Are you targeting one first and then the other? And how does that impact, for example, things like the endpoints if you were to move that into your clinical trial? So I think another consideration we're thinking about even the trial readiness and those early studies is what do you know about the disease, about how perhaps certain elements of the disease might impact safety assessments. Also, what do you know about the endpoints? And so I know people are always being asked to be involved in natural history studies and registry studies, but there is so much value in those because they really help you to set the stage for hopefully successfully designing the trials and taking it to that next phase in clinical development.
Mike?
Yeah, no, thanks. So I think my panelists covered quite a lot already, but I think I would just add a couple more points that, and it kind of goes along the lines of, I think, what a lot of folks have already said, which is, I think from my perspective, the presence of good animal models is always really important, and not just for gene therapeutics, but for really drug development in general. And I think one of the advantages that we have in CMT drug development or gene therapy development is that those models do exist, and I think that's that's promising, and that really does set the stage for being able to translate all of the good work that's been done for many years in the non-clinical space into the clinic. I think some of my colleagues already touched on the clinical development pathway, so that is obviously something that we look at quite closely. We're fortunate that the INC and other organizations have already pioneered a lot of the infrastructure, have put together natural history studies, but obviously understanding what are the endpoints, what are the outcome measures that not only matter to patients but to regulatory agencies. That's very important. And what does it take? What is the size and scope of the clinical trials that will be required to show that a therapy is effective? I think those are all some of the additional things that we look at in clinical trial development with gene therapy and others.
Cleopas, do you want to chime in?
Yeah, I think most of it has been said. Just to recap, we need valid mechanisms. We need to understand the disease. We have to demonstrate efficacy in the models that are, appropriate, and we need to demonstrate safety. And companies also want to make sure they will have a landscape when they come to design clinical trials where the natural history data will be there. Patient cohorts will be accessible, so they will have enough number of patients to reach out to test treatments, and also they have also a consideration of the market later on. That's something that I learned when talking to them. They want to know also whether this will be a treatment that will stabilize the disease or reverse some of the symptoms. So they have some considerations from the business point of view, but certainly what I outline in the path to clinical development, that's all points that the industry and we also have to consider in everything we do.
Thank you. So many of the panelists have already referred to cases where there are really spectacular successes in different types of gene therapy and many modes of gene therapy that are either approved or in clinical trials. So with the experience that many of the panelists have, the next question is really, how do scientists and biotech companies take learnings, lessons learned from gene therapy efforts in other diseases, and apply those to CMT? And maybe I'll scramble the order here a little bit. Maybe I'll ask Mike to chime in first on this one, if that's okay.
Yeah, sure. No, happy to. So, John, I think our experience with gene therapies and other diseases, it is very important to biotech companies. Even though different diseases are different, but I think, for example, there are now gene therapies that have made it all the way through the clinic and onto the market in other genetic devastating diseases like SMA or spinal muscular atrophy, TTR polyneuropathy, or hereditary transthyretin neuropathy. And I think what this shows is that there is a viable path in the clinic for these types of therapies, which not so long ago were thought to be sort of quite novel and quite experimental. I mean, they still are, but I think the point being is that now there is a path that's been paved by other researchers and developers that I think we in the industry follow quite closely and try to learn from. I think even unsuccessful trials, I think, are also really important, and they don't necessarily discourage us from moving forward, because you learn so much from both a successful trial as well as a non-successful trial. And so I think we from my part, I can say that we look very closely across different indications for sort of guidance on how to develop gene therapies in CMT.
Maybe I'll ask Diana and then Catherine after that. So go ahead.
Sure. So yeah, I think very similar. I think the real value in those lessons learned from other studies or from even approved therapies really is both to understand from the clinical and efficacy perspective, the design, the effect size, kind of to help instructing trials and endpoints and design. But a key component as well is really the cumulative knowledge about safety, especially for the gene therapies. I mean, we're still, I think relatively early in this field, and yet showing a lot of promise, especially for our, neuromuscular disorders and for the neuropathies. But what can we learn about, the tolerated doses and or route of administration and the pros and cons of those routes, as well as even for the approved therapies, really think, constantly evaluating, safety signals and how that instructs, for example, added safety measures, perhaps that need to be included in prospectively in clinical trials. And so I think not, thinking about, studies for rare diseases in silos, but really thinking about the cumulative knowledge gained by every one of those studies and being able to share that data to help instruct and really maximize the safety and how we develop studies in the future, and how we monitor the individuals who receive these.
Catherine, Go ahead.
Yeah, I think, very important points already made, especially how to handle side effects that can occur. And I'm actually excited to see that people are very willingly sharing this information, which is extremely helpful. I think we've learned a lot about dosing for different injection routes, but then I think one aspect that hasn't been brought up is also the manufacturing. We're getting better in making viruses, we're getting better in the analytics to be more exact, to really know what kind of doses, what are the ingredients that we are giving to the patients. And that also helps making these treatments more safe. And another thing that I would point out is also the standard of care, understanding that a lot of these diseases have multiple aspects, systemic aspects, nutrition, physiotherapy, that can further also help improve some of these therapies and have to be, really taken into consideration as well.
So, Luke, maybe I could make this question a little more specific. There's a lot of excitement about CRISPR-Cas9 and gene editing, and it's very exciting that some of these things are moving through clinical trials on their way to approval. And so in terms of learnings or lessons learned, for specifically applied to CRISPR-Cas9, I wondered if you could comment on that.
Yeah, absolutely. I think we're learning a lot from the other studies in the same sorts of things that have been alluded to already, but that are specific to gene editing, specifically the sort of risks, the side effects, and what are those safety and monitoring studies that we need to do both before starting studies in patients and also to monitor those patients after the treatment has gone on. And so I think just from a very general standpoint, what's going to be really important for us is hopefully seeing not just positive results in terms of helping with the disease, but also showing that this approach can be safe, and seeing the kinds of testing that people are doing to make sure that we're not introducing other unwanted genetic changes that might lead to things like cancer, or other side effects that are toxic and harmful to those patients, even though they're treating a different disease, is going to be incredibly important. And also because a lot of these approaches are using slightly different versions of the CRISPR enzymes that modify the genome, we're going to be able to learn more about how those different types of tools play out in the human body, and get a lot of knowledge from that, which can help inform us about the way we can select those variants going forward.
Cleopas, do you want to chime in on this overall question?
Yeah, I think it's very important to have a very careful safety evaluation of any approach. I think that's key, and definitely we can use knowledge obtained from already clinically applied gene therapies for other diseases. A lot of this knowledge can be transferred to CMT neuropathies. Some of the tools we use are actually used for other diseases and have reached clinical stage. So I think that will accelerate the process. But we have to be very careful because CMT neuropathies are not life-threatening diseases. And we're not talking about disorders of infancy in most cases that are very rapidly progressing. So we need to consider other factors, but definitely all experience with gene therapies that reach the clinic already will facilitate the progress also in CMT gene therapies.
Good. The next question that we have is really the interest of the community as a whole, obviously, to know when these things will show up in clinical trials and as approved therapies. And so I guess what the question is how do we set these timelines and the expectations for those timelines? And I wondered if Mike would maybe kind of comment on sort of the general process involved in setting timelines to get to clinic.
Yeah, no, happy to. And I think, Cleopas, highlighted a little bit of that, the process to getting a drug, all the way through to approval at the outset. But I think that, the good news is that I think we're very close to having, therapies enter into the clinic, in the gene therapy space. And I think a lot of this is really because the infrastructure for clinical trials has been put into place, and a lot of research has already gone into that. Typically, once there's sort of non-clinical proof of concept, typically in an animal model, there's obviously still a series or battery of safety and pharmacology studies that have to be completed in preclinical models. That can sometimes take a year or two to complete before there's an adequate profile in terms of the pharmacokinetics, the safety, the predicted therapeutic and safe margins in humans can be established. And usually once that's been defined, then a company such as ours would put together something called an IND, or Investigation of New Drug application, which is that's typically submitted to the FDA. And in other countries, there's the equivalent of that also needs to be submitted. And what that does is that signals to the regulatory agencies that we believe we're ready to begin human trials. Once that is approved, then we initiate our trials, our first in human trials. Traditionally, in many different indications and disease states, drugs go through sort of a phase 1 to then phase 2, phase 3 process, kind of in that sequential manner. But I do think, in the case of CMT, with genetic medicines, oftentimes there are opportunities for I guess more smart development or streamlined development, partly because this is considered a rare or orphan indication, as is often designated from a regulatory standpoint. And so clinical trials can be somewhat fewer and smaller in size, and still be acceptable for sort of proof of substantial benefit, right? I think some of the advantages that genetic therapies have are when you are targeting the underlying disease, underlying defect, there are certain markers that are, I think, highly relevant. Gene expression markers and other biomarkers can be looked at early on, I think, to give you a pretty good sense of where the safe and effective dose or dose ranges are. And sometimes we'll take advantage of that and move a little bit more quickly, perhaps initially to patients with disease rather than healthy volunteers, just because of the mechanism of action. It is more of a streamlined path in that regard than in some of the more traditional medicines and larger disease indications. Probably not fast enough for a lot of our patients, but I will say that it is I think a path that can be more streamlined in this indication.
Excellent. So I think the next question, maybe I'll direct to Cleopas first. There's a lot of interest in the community about how people can participate in clinical trials, and I wondered, from your perspective as a neurologist, what do you think are likely the age range that's anticipated for first clinical trials? And I know this answer probably varies by the type of CMT, but I just wondered what your perspective was.
I already mentioned before, we have to consider that these, in most cases, are not life-threatening diseases. They progress very slowly, so they get worse slowly over time, and most patients start having symptoms in late childhood, adolescence, and early adulthood. So I believe from the regulatory authorities' point of view, it would be unlikely that they will approve testing first treatments in minors. And I believe that the first testing in the clinic will be in adults. I would assume that would be 18 to 60-year-old range. In this range, at least for the most common forms, we do have already longitudinal data, and we have some clear idea about the rate of progression, which can be predictable based on past experience and past trials. So I think this will be the age range that we should target at first. Of course, we all know that as time goes by, there is more damage to the nerves. And in ideal world, we want to treat as early as possible, even in children, because then we will prevent symptoms and progression of the disease. But I think this will be in a second stage of the whole effort. That would be my prediction. But again, it depends on the type of the CMT. Some are much more severe. They affect children much more, and some are milder and slower in the way they develop. But I think overall, I would see the first testing in adults.
I might ask Diana the same question, partly because I know you have experience with some more severe forms of CMT as well. So.
Yeah, so I think I agree. It depends on the severity, really, and really the subtype. In terms of what's that trajectory. I think with a more severe, infantile onset or childhood onset disease, that window by which, where you probably have the maximum effect or, potential prospect for benefit while the nerves are still healthier, I think does push you to consider trial designs where you are thinking about children and even, very young children. So it really does boil down to, of course, the condition and what's really the age of onset. For conditions, though, perhaps where there could be variability, and you do have even a later onset form, or for the types where classically they present in kind of adolescence to adulthood, then I completely agree. I think thinking about if you have that opportunity for those conditions to start in adults, but really thinking about once you've established some markers of safety, really thinking about then can there be an additional trial built in based on that safety learned and some of the early efficacy to then bring those down to also the pediatric population. So it really does, of course, boil down to the type of disease and really how severely it progresses, and also where you want to intervene so that you're not missing that window to be able to really see the optimal benefit.
Well, thank you so much. I think with that, we've reached the end of our time here. I think from the perspective of the CMTA, we're very grateful that we've been able to benefit from these very talented and experienced people that have been on this panel that are actively working in this field. And I really appreciate their time today. I know there are some questions that maybe haven't been answered directly from the audience, and we'll do our best to answer those offline as well. So again, thanks so much to the panel. It's really been a great discussion.
Thank you.